Hello everyone, my name is Peiyuan Feng, a PhD student from Tsinghua University, Institute for Interdisciplinary Information Sciences. I will give a talk entitled Predicting MHC-peptide Binding Affinity by Differential Boundary Tree. As we all know, in an immune response, endogenous antigens derived from the mutant proteins or infect virus are digested into linear peptides. These peptides bind to MHC-1 molecules, resulting in MHC-peptide complex, which is present to the surface of the cells. Then, cytotoxic T cells recognize infect or cancer cells by the binding of T cell receptor and MHC-peptide complex. In this process, the binding of peptide and MHC-1 molecules with high affinity plays an essential role in antigen presentation. In humans, the MHC molecules is encoded by HLA gene family with thousands of variant alleles. These alleles bind to a large variety of antigens, which guarantee the diversity of immunity. Our task here is to predict the binding of peptides to individual alleles, and in the last decades, lots of computational methods have been developed in solve this task. These methods can be mainly divided into two categories, allele-specific and pan-specific. The allele-specific models usually take only peptide sequence as input to train one model for each allele, while pan-specific models take both peptide sequence and MHC pseudo sequence as input to train a single model for all alleles. However, these methods still occupy several drawbacks. First, they suffer from a high false positive rate of predicted epitopes, and most of the deep neural network-based methods fails in identifying single amino acid mutations that may significantly influence the binding affinity, which is important for subsequent design and sequence optimization. Moreover, the deep neural network methods cannot provide interpretable understanding of model prediction. In our study, we propose a differential boundary tree-based model named DBTpred to solve these problems. Boundary tree is a special type of k-nearest neighbor method. It selects a subset of data instances to represent the whole training dataset by organizing this instance into a tree. Each node in the tree represents a training instance, and the properties of boundary tree is that the adjacent nodes in the tree are the closest to each other, while the labels are distinct. For example, in our task, two adjacent nodes in the tree are the MHC-peptide complex with similar sequence, while their binding affinity is different. Once we have a boundary tree, we can query this tree to find the closest node of the test sample. For example, when query node enter the boundary tree, it first reaches to the root of the tree, which is said to be the current node. And then we compare the query node to the current node and its children. We update the current node to the child node that is closest to the query node. And this process continues until the query node is much closer to the current node than any of its children or reaching the lift. We can assign the current node as the closest node and then transfer its label as the prediction of the query node. When a query node finishes the traversing process, boundary tree can generate a decision path for the final prediction. After identifying how to query a boundary tree, we can build a boundary tree in an online manner. So here is the procedural course of this algorithm. Suppose we have a training dataset. We first randomly sample a training instance as tree root. Then, for every randomly sampled training instance, we traverse it to the current tree to get the closest node in boundary tree. Next, if the label of training instance is much different to its closest node in the tree, we add this training instance as a child of its closest node. Under this process, we can maintain the property that for any adjacent node in the tree, they are similar to each other while their labels are distinct. The key element of boundary tree is the distance between training samples. Intuitively, we can directly use some distance measurements of sequence data, such as eddy distance. However, we can take advantage of strong power of deep neural network to learn the distance measurements between arbitrary two examples. More precisely, we take both peptide and MHC pseudo-sequence as input to a well-developed neural network and output the feature vector of these samples. We calculate the Euclidean distance of two feature vectors as their distance. And here is how we train the neural network together with boundary tree in a two-step manner. We first sample a mini batch examples from training data and build a small boundary tree using their feature vector. And then we sample another batch of training data and use this data to query the boundary tree to get the predictions. Specifically, once we get the closest node of the query sample in the boundary tree, we assign the weight average of labels of the siblings of closest node as the final prediction to query sample. The weight score is calculated by the normalized distance between query node and sibling nodes. For each query node, we calculate the gradient based on the mean square error between predicted scores and its labels, and then update the trainable parameters in deep neural network. This process continues until the training loss converged. However, this training process is very slow. Since for each query sample, we need to traverse the query sample one by one in the boundary tree and build a new computing graph to calculate its gradient. In fact, it is impossible to use these training algorithms in large datasets. To solve these problems, we propose a parallel query algorithm to accelerate the training process. The purpose of this algorithm is to query a batch of nodes simultaneously. First, we decompose the intermediate nodes of the boundary tree into different subtrees, where each subtree contains one intermediate node and all of its children. Then, we construct a query matrix Q, in which rows represent query samples within the same batch and columns represent all the subtrees from the decomposition. For each query node, we search for its closest neighbors in each subtree based on the Euclidean distance of neural network outputs. The closest neighbor is labeled as 1 and otherwise as 0 in matrix Q. For instance, suppose that the closest node of S2 in the three subtrees are N2, N4, and N5. Then the values corresponding to the N2, N4, N5 columns are labeled as 1 in row S2. This comparison only needs matrix manipulation that can be fully parameterized on different computational resources. Next, we construct a traverse matrix H, in which rows represent subtrees and columns represent all the paths of boundary trees from the root. For each edge in the subtrees, we assign a well-designed score to it. Then, we propose that for arbitrary query node Si, its traverse path is the path with the maximum index when Si row multiply with Hj clone. More specifically, when multiply Si with the matrix H, we in fact calculating the summation of scores for each pass. And through the clever design of matrix H, we ensure that the traversing path of Si occupies the largest score summation. The score design and the proof of this algorithm can be found in our paper. Using this algorithm, we can transfer the path search process into a matrix multiplication problem, which can be easily pivotarized through using GPUs. We compare the running time between original query algorithm and our parallel version. You can see that the running time of the original algorithm is linearly correlated with the number of test samples, while our parallel algorithm requires constant time. We had to clarify that the parallel algorithm requires lots of memory when size of boundary tree and the number of test samples is large. The Q and H matrix is very big. However, in our study, it's acceptable that usually memory usage is less than 40 GB. When building the boundary tree, we ensure that the absolute label difference between the query node and its closest node was larger than a threshold epsilon. We found that the epsilon should be smaller than 0.1 to ensure the model with good training performance. It's not surprising that the larger the epsilon is, the less nodes left in the tree. So when the epsilon was too large, the performance shifted a lot, since there were less samples in the boundary tree to represent the whole dataset. In addition, the ratio of the number of the tree nodes to the total number of training samples decreased as the number of training epochs. This result indicates that the training process enabled the model to learn good feature representation. Finally, we noticed that the DBTpred only need 40% training examples to achieve over 0.8 test performance in terms of Pearson correlation coefficient. We further evaluate the performance of DBTpred. We conduct a 5-fold cross-validation and compare it to the state-of-the-art method MHC-3 and ISMB. We found that in almost all lengths of peptides, the DBTpred outperformed these baselines. In detail investigating each allele performance, we found that in most alleles, the DBTpred achieved better performance compared to net MHC-3. And to further demonstrate the generalizability of DBTpred, we test its performance on an independent MHC class 1 binding affinity benchmarking dataset, which contains over 30,000 MHC class 1 binding peptides. And DBTpred still outperformed all the other benchmarking methods. Intuitively, the ratio of samples that left in the tree to the total number of samples represent the learning complexity for each task. Here, we investigate how many samples are generally needed in DBTpred for each allele. And we found that through the ratio of samples left in the tree, we can classify the learning tasks into 3 categories. The first one is the well-learning allele, which achieved high performance and represent by only 30 to 40% of the samples. Most alleles located in these categories. And the second one is poor learning allele, with poor performance and high ratio of represented samples, which indicates that these allele tasks were not properly learning And the third one is easy to learn allele. These alleles obtain high performance while with low represented samples. And when investigate the label entropy of these alleles, we found that they are relatively small, which means that there are lots of same label in these tasks. And we found that in fact, most labels of samples in these alleles are zero. Another application of DBTpred is that it can provide a decision path. And here is a simple example of the interpretation of how DBTpred predict a test sample. In order to demonstrate the important residues that influence the sample distance, we calculate the silencing map in terms of the Euclidean distance between the test sample and each tree node. DBTpred clearly demonstrates how the test sample traverses to its closest node step by step. For example, the model first searches for the last value in the peptide, which occupy the largest silencing map score. And then it searches for the cysteine and 2 valence in the first, fifth, and the last positions of peptide, and get the closest node. These 3 residues are the key residues in determining the prediction of binding affinity of the test samples. And the path also displays several possible point mutations, which may strongly alter the binding affinity. And this information may provide useful clues for the following sequence optimization. And finally, here is our conclusions. We developed a differential boundary tree algorithm to predict the MHC-peptide binding affinity. We propose a parallel algorithm to accelerate the training of DBTpred. And we found that DBTpred achieves better performance compared to the other state-of-the-art methods. And the statistical properties of the DBTpred indicates the complexity of the learning tasks. And our model can provide interpretable decision paths for each predictions. I'd like to thank for the enlightening advice from Professor Zheng Jianyang and Professor Ma Jianzhu. And here's my presentation. Thanks a lot.